Hello, everyone, and welcome to the continuation of our lecture series on the excretory system. So we're going to talk about a couple more organs in this system, and then we're going to move on to various diseases of the excretory system organs, and then we're going to talk about the biomedical devices, recent innovations and implants that are used to treat some of these diseases. So last session, we actually started talking about the function and physiology behind the system, and we also discussed the skin as one of the main organs. Today, we're going to continue, and we're going to talk about the lungs, our kidneys, and the liver. So hopefully today we'll be able to conclude, and then in the next lecture, we're going to talk about various diseases in the excretory system, and also we're going to talk about the mechanics and physics of the excretory system functions. So without any further ado, we're going to start today's lecture talking about the human lungs. So as you probably know already, we've got two lungs, the left and right, and they're fed by the right ventricle. So the pumping action of the heart, as we've covered it in the second portion of this module, starts from the right ventricle, where the blood is pumped to the lungs, and then it's circulated back through the left atrium and left ventricle, and then it's actually pumped to all the other organs through the aorta. So in this cycle, as you can see, the lung and the heart are very tightly connected, and it's basically also that way when you look at it physically. So the lungs are wrapping around the heart, and therefore the distance between the two in that pulmonary artery region is very short. And so what happens is that as the blood exits through the pulmonary valve, it enters the lung, and then after going through the alveoli and the smaller capillaries, getting all the oxygen in, it's going to then come back to the heart through the the mitral valve, and then it's going to exit finally through the aortic valve. So that's really the circuitry which we've already discussed before, but now let's try and see what's going on inside these massive structures. So if we look at the lung in a bit more detail, we'll see that starting from your nostrils, you actually have the entrance of air, where they're actually the molecules and the stream of air entering is going to be filtered using the hairs in your nostrils. That's the first level of filtration. Then this flow basically goes through the nasal cavity and through the pharynx, and the pharynx is going to then navigate it towards the trachea, which is this massive windpipe that you can see here. It's a pretty large diameter, very much like the aorta. It's a couple centimeters. And the trachea is then going to divide the flow into the two bronchi. So these bronchi are then going to branch down into bronchioles, and these are the smaller branches, just like arteries and arterioles in the aorta. Here we've got the trachea, the bronchi, and the bronchioles. And then what happens is they, towards their end processes there, they're going to circulate through a small structure called the alveoli, okay, which are basically the air sacs. And then they're going to circulate back. So the vasculature is basically going to wrap around these airway structures, as we'll take a look at it in a minute. So the lungs are basically coated with epithelium. So these structures that you actually saw that start from the bronchial and the bronchioles and the bronchi, they go all the way down, and in all this airway system, you have a coverage of epithelial cells, which form an epithelium, very much like the endothelium that coats the vascular structures. The epithelium coats these airway ducts, if you will. And what happens is that the epithelium lining acts as a membrane, as a trap for all those extraneous particles such as dust and pollen and the other contaminants. And so this filtration system is as such that you have the oxygen, which is able to then pass through these membranes or these cellular walls into the blood vessels that wrap around them. Again, if you look here, you'll see the structure of the alveoli and the blood vessels wrapping around the air sacs at super close proximity. So if you look at the walls of these alveoli, you'll see that they're only a few microns. And so there's a very short distance between the air inside these sacs and the blood running through these capillaries. That's how you get an exchange of oxygen through these walls. Now, what happens is this cluster of sacs gets inflated when the air comes through the bronchioles, and then it actually expands and basically expands the capillaries as well. And so this oxygen inrush is actually going to diffuse through the wall and oxygenate the blood in the capillaries. And the other thing that happens, which is as critical, is the CO2, carbon dioxide exchange. And that's when the carbon dioxide from the blood that's come from all the way from the other organs is now trying to exit through the air sac, and then the blood is trying to get replenished with oxygen and depleted from its CO2 so that it can go back to the heart and then get pumped through the left ventricle. So that's really the two main functions of the lung, is the exchange of oxygen and carbon dioxide. And in all of this, you have the epithelium as that membrane. So the diffusion through the epithelium is very critical, as we will study diffusion through hydrogels, for example, in our lab, in the third lab. You'll see that diffusion is determined based on the molecular structure of the medium. And in this case, it's actually a much more complex situation, because you've got cells which have their own multiple layers of diffusivity coming from the cytoplasm and the cell membrane. So it's a very complex problem to analyze this, really, or model it. That's why we usually use in vitro models to simulate the exchange of oxygen and CO2, and to understand the formation of the epithelium and the endothelium. So in terms of the macroscopic physiology of our lungs, we have a constant change in volume of the lungs. So if you look at this plot, at first sight, it may look a little bit weird, because so this is showing the progression of volume as the volume of air in our lungs is changing. You may wonder why we suddenly have a strong peak or trough, whereas, you know, as we breathe naturally, we're just having this cyclic situation. Well, the reality is that we actually sometimes take a deep breath, and then sometimes actually exhale deeply. So what we call this is a forced inspiration or forced expiration. And the idea here is that you are trying to push out all the air in your lungs, or you're trying to take in more air, as much as you can. So this plot is talking about the maximum amount of air you can take in and the maximum you can push out, okay? So if you try your best to push out as much air as you can, you'll reach this minimum possible volume. okay? Uh, and then if you try and take in or inhale as much air as you can, the maximum that you can reach is this peak up here. But usually you're operating in this region, okay, which is your natural breathing cycle, where you're moving between a minimum and maximum air volume in the lungs, okay? So what happens is, this is the same for all mammals, is that we have a negative pressure pulling in the air, sucking in the air, and the volume of our lungs increases as we do that. And so this volume fluctuation that you see in normal breathing is a tidal volume, okay, which is the amount of air that you typically inhale and exhale. Now, the vital capacity is this distance between the maximum and the minimum volume, which is the maximum range, in other words, that the lung can go from the least possible amount of air inside it to the maximum amount possible. This is the vital capacity. And basically, we have the inspiratory reserve volume, which is when you're doing the inspiration, and then you have the expiratory reserve volume when you're trying to expirate as much as you can. So if you actually look at these oscillate around a positive volume. So our lungs never go to zero, okay? They don't collapse completely to get to zero volume. So they're always positive, and they're fluctuating between a certain volume, a certain minimum, maximum volume. But then, as I mentioned, you can take a deep breath and expand your lungs to the maximum possible, or you can actually try and exhale as much as you can and get it to the minimum possible. But you cannot go below this, because otherwise it will actually damage. This is the residual volume, so that you can't really go below this. This is a residual volume. This range here is a tidal volume, and this range between the two is the vital capacity, as you can see here. So this is really the macroscopic physiology and physics of it, and we will look at more depth into the mechanics of our lungs when we actually study the alveoli sacs in a bit. Now, moving on to the next organ in this system, we've got the liver. And when we're talking about the liver, we're talking about a pretty similar structure in terms of exchange of nutrients or waste, small molecules, through these epithelial walls that line the cells. And so basically, it's a very similar situation. Here you have a bile duct, and then you have the vein, and then again you have exchange between blood and between the bile fluid here. So it's very similar to the air-blood interface and exchange that happens in the lung. And in the case of the liver, we're talking about proteins, okay, the excess proteins that arrive at the liver to get broken down and decomposed. And then these smaller molecules, the amino acids, are then broken down all the way to form ammonia, which then converts to urea and is carried to the kidneys to then leave the body. So it's really another level of turning the food that we digest and turn into proteins into amino acids, and then, as I mentioned, ammonia and urea. So what happens, the cycle is really that urea then goes through to the kidneys, and this main function of the liver is doing this reaction, this chemical conversion, all the way from the proteins to urea. And then finally they get filtered out and excreted in urine. Another function of the liver is to detoxify blood, meaning that it filters these substances and bacteria and drugs, and it actually changes them into less toxic forms. And finally, another function is to excrete bile. So basically, apart from the formation of urea, which is a chemical conversion there, detoxifying blood, it also excretes very much like the other excretory organs that had excretion and secretion. And what it excretes in the bile fluid is the worn out red blood cells, the salts, and cholesterol. So this is really what the liver does, and it has this lobular structure to allow for optimum contact with the blood that comes through the central vein and divides through those branches, what we call the branches of the portal vein. So these, as you can see, all of these excretory organs have pretty much the same blood media contact architecture in various shapes, but the same physics. And finally, we have our kidneys, and our kidneys also have a very similar structure. Now, this time, instead of having the alveoli, or in this case, those ducts that you have, the bile ducts, you have the nephrons here. And the nephrons are going to be those units that exchange those molecules and ions with blood. And what they do is this process called osmoregulation, which is the control of water concentration in the bodily fluids. It's interesting to know that the kidneys actually use a lot of energy. They use about 25% of the cardiac output. And they similarly, they do the removal of waste from blood, as the other organs are doing to some extent, as the lung is doing in the form of CO2. They do it in the form of ionic and protein exchange. So they deal with that urea that I mentioned that came from the breakdown of the proteins in the liver, and they also deal with the other salts and ions, and they excrete a significant amount of urine, about 1 to 2 liters a day, and that's also in charge of regulating the hydration of the body. So therefore, they need a very accurate control loop to make sure everything, the osmolarity of the blood and the fluids, the concentration of various ions is there. So they do lots of reabsorption as well, very similar to the other structures. And finally, to look at this and sort of tie it to the circulation network, the renal arteries, we've looked at these before in the cardiovascular section, are what bring the blood through to the kidneys and then drive them out through the renal veins. And what happens is that these renal arteries that bring the blood goes to collect in the basically the ducts and the pelvis, and then this is the site where it gets absorbed. So the nephrons basically are the small building units of the kidney, and if we look at their structure in more detail, you'll understand how this blood circulating from the renal artery is in close, wraps around the tubule there, and this tubule is basically carrying the fluid that will be then secreted, and so the blood is in super close proximity of that, allowing for diffusion of those species to that biological fluid that's going to then be disposed of through the urinary tract. So basically the liquid goes through the two sides of this the two limbs, and then travels all the way to get basically through the urinary tract. Now, we're not going to look at a lot more detail into this, but this is mainly to give you an understanding of what they do at the cellular level. You've always got a very close interface, as I've repeated many times, between the blood vessels, and the secondary media, the fluid that's being circulated in that organ. That's pretty much a signature of all of these organs. Okay, thank you very much so far for listening in onto this section, which we've sort of taken forward up to the physiology of these individual organs. In the next lecture, what we will be looking at is the mechanics of the excretory system and the excretory system in health and disease. And after that, very soon we'll get to talk about the biomedical devices and materials used in the excretory system and the implants that could be used. Or we'll also try and look at extracorporeal biomedical devices, which are also going to be quite interesting. So let's take a quick break and then continue on with those sections. Thank you.